right, good morning everyone. So before I get started, I want to first thank Melinda and the CRB for giving me the opportunity to speak to you all. And so today what I'm going to tell you about is how work in the core breeding program at UC Riverside is being done to develop genetic tools to facilitate new variety selection. All right, so to get everyone in the right frame of mind, I want to start with this question. So how long does it take to develop and release new citrus varieties? And so to illustrate this point, I'm going to use two commercial Mandarin cultivars as an example. So the first is Gold Nugget. So the cross to generate Gold Nugget was performed in 1955. So just to put this into context, this is the year the film Rebel Without a Cause was released to movie screens across the US. So a whopping 45 years later, this variety was commercially released in the year 2000. And so this is the same year that Gladiator graced our movie screens. So you can see quite a lot of time has gone by in this scenario. So this is a worst case scenario or worst case situation. And so the next cultivar I'm going to show you is one of our best case examples, and so I'm going to use Tango to illustrate that point here. So the time it took for Tango to go from this initial event, which was a mutation event, to release was only 11 years, and so this is relatively fast in comparison. But I just want to highlight the fact that this was enough time for the entire Batman franchise to experience a reboot. So this is quite a lot of time still. And so, to illustrate this further, I want to highlight some of the major phases of our breeding program or breeding cycle here on the bottom of the slide. So, it all starts with an initial cross between two promising parents. So, seedlings from that cross, usually about 100 or so, then enter a preliminary evaluation phase in the field. And here's where we identify and select the most promising hybrids that have the best suite of characteristics that we're interested in. So these individuals then enter the CCPP to develop a disease-free source of material. And this material is then replicated in field trials, often in many locations, to ensure that those characteristics are robust. And so if all goes well, the variety is then released for commercialization. And so you can see that the time that it takes to go from start to finish in this process is 15 years. And so even in the best case scenario, you can see it's a career's worth of work to see this through from start to finish. Okay, so what strategies can we use or have been used in the past to optimize this process? So of course, we can decrease the overall length of this breeding cycle. And so many decisions over the years have been made in order to minimize the time that any hybrid spends in each of these particular phases. And so until we can reduce the amount of time that it takes for an individual to produce fruit, we don't have much wiggle room in this area, but maybe you'll hear a little bit more about this afternoon. Okay, so an alternative strategy is to increase what I'm going to call the efficiency of hybrid selection. And so this is really focusing on the early parts of this pipeline and ensuring that any hybrid that enters the preliminary evaluation phase will be desirable in some respect. And so this increases the probability that it will be of commercial interest by the time it reaches the end of the pipeline. So of course, technological advances, particularly in genetics and genomics, now allow us to really address this issue of increasing efficiency of hybrid selection. And so we address this using three main aspects. So the first is what's called trait discovery. So we use genetics to understand what genes or regions of the genome underlie important traits that we're interested in. The second is what I'm going to call hybrid generation. So with this information in the future, we can identify parents to cross that will produce desirable progeny before those crosses are even made. And then lastly, we focus on what's called trait selection. So we use markers or genetic information to identify and select those most promising hybrids to enter the pipeline. And so today, like I said, what I'm going to tell you about is work by the core breeding program that has developed resources in this field in order to address this issue in the early parts of the breeding pipeline. Okay, so success at each of these levels depends on developing a deep understanding of the relationship between genotype and phenotype. And so what I mean by genotype is illustrated in this cartoon here. So I mean the unique DNA sequence of an individual tree. And so in this cartoon, there's 6 individuals. You can see that the sequence is identical at every position except for this A to T variant, which I've highlighted in gray in the center. And so these variants are the portions of the genome that we're really interested in. So by phenotype, what I'm referring to are the physical characteristics of the tree. And so in this image here, you can see that the tree on the left is diseased, while the tree on the right is healthy. And those are the phenotypes of those 2 particular trees. Okay, so identifying the best hybrids requires genetic information. And so I've only been at UC Riverside now for 2 years. And luckily for me, this information was developed before I even arrived. And so I'm not going to go into the details of the platform or method that was used to do this. But I do want to highlight the fact that we have genetic information at a genome-wide level for over 2,000 individuals. So this includes individuals in the germplasm collection or the citrus variety collection at UC Riverside, as well as important breeding parents. And so this data was produced by Mike Roos and his group using a combination of USDA and gift funds. Okay, so this genetic information is essential for our work, and it vastly increases the efficiency of hybrid breeding, as I've outlined to you all so far. Okay, so how can we use this information? So I'm going to highlight one way with this slide here. So this genotype data has already been used to increase the efficiency of the breeding program by 150 or to 200%. And so how is this done? So genetic markers are used to distinguish what's called zygotic seedlings, which are true hybrids, from nucellar seedlings, which are clones of the maternal parent. And so we certainly don't want to evaluate clones of a maternal parental variety that we already know its performance in various situations, and so we want to remove these before they enter the breeding pipeline. So in the image here, I'm showing you an example of some hybrid and nucellar seedlings from a cross between a mandarin and a grapefruit parent. And so in some cases, we can use phenotypic markers, like in this case, to identify true hybrids, which are on the outside of the photo, from those that are nucellar. But in many cases, we don't have these visible markers. And so starting in 2017, every year, 1,000 to 1,500 seedlings are screened to identify those true hybrids and remove those that are nucellar. And so this has had 2 benefits to the program. So it's expanded the repertoire of female parents that can be used, and then it allows us to cull or remove those nucellar seedlings, which can occur at a rate of 50% to 97% in any specific cross. And so with genetic data, there's been vast improvements in the efficiency of the program by identifying these true hybrids that we're interested in. Okay, so now we have extensive genetic information on these lines, but there's still some areas where there's gaps. And one of those gaps is that we don't have large phenotypic data sets in citrus. And so I'm going to tell you a little bit today on work that we've done to close this gap. So there's, of course, a number of challenges with phenotyping citrus. So this includes the fact that it's labor intensive, which means that it's, of course, expensive. The long juvenile period in citrus makes it difficult to evaluate traits that can only be measured in adult trees, like fruit, for example. And then there's also, of course, environmental effects, which makes disentangling this relationship between genotype and phenotype difficult. Okay, so luckily for me again, there's been advances in this arena over the last 10, 15 years. And this has to do with the fact that now cameras are much, much cheaper than they used to be. And so I'm illustrating that with this plot here. So now each and every one of us is carrying around a relatively cheap high-resolution camera in our pocket. So this is your cell phone camera. It costs about $10 now. And so these cheap cameras have been applied to a number of challenges in plant breeding. And so with imaging, we can now more efficiently capture and measure traits that are of interest and that were not obtainable before this. Okay, so today, or I should say that the work in our group is focused on using imaging to collect phenotypic data in citrus using two main platforms. So the first is the commercial pack line that's available to us at Linco Research and Extension Center that many of you may be familiar with. And this we're using to image a very large number of fruit and extract information on traits that are of interest to us. Secondly, we're using cameras attached to drones to image orchards, and from these images we can measure traits like tree size and health, among many others. And so this is being applied not only to the germplasm collection, but also to field trials that are located around the state. Okay, so today I'm going to focus on work in the core program that aims to understand what genes underlie traits that are of interest using some of this imaging data that I just discussed. All right, so the commercial pack line facilitates the genetic analysis of fruit traits. And so this piece of equipment was designed, as you know, to separate marketable fruit from fruit that has some kind of damage and is unmarketable. And the way that it makes this decision is by taking 30 images of every single fruit, imaging in a variety of spectrum, and from those images we can extract traits related to fruit size, weight, shape, texture, color, as well as other defects that may be of interest. In addition to using the pack line, we also collect information on Brix and acid and seed number using destructive fruit quality methods. Okay, so I want to remind everyone that there is an immense amount of genotypic and also phenotypic diversity in the germplasm collection at UCR, in the citrus variety collection. And so I'm trying to illustrate that with this wonderful image here. So here's a sampling of fruit from that collection, and you can see that there is an immense amount of variation in fruit color and shape and size among many, many other traits. And so what we're doing is we're trying to use the pack line to get at what is the genetic basis of these differences in order to use that for breeding cultivars in the future. And so how are we doing that? So this past season, we harvested over 60,000 fruit and ran these fruit across the pack line at Linco. So these fruit were sampled from 600 varieties in the citrus variety collection. And these varieties were selected to represent known genetic diversity in that collection and in citrus and its relatives in general. And for each tree, we sampled about 50 fruit to get a good estimate of the characteristics associated with that tree. Okay, so what can we do with this kind of data? So again, I want to remind everyone that citrus trees, or the experimental trees that we work with, are grafted. So this means that the genotype of the scion, or the above ground portion of the plant, is different from the genotype of the rootstock. And so with this data, we can begin to understand what is the relative contribution of scion and rootstock genotype to the variation in some of these traits that we've measured. And so the answer to that is shown in this plot here, and so I'm going to walk you through it quickly. So on the x-axis, on the bottom portion of the graph, is all of these phenotypes that we've measured from fruit. So size, shape, color, Brix, acid, etc. On the y-axis is the amount of variation that the scion genotype explains for those traits. So this is going to tell us how much genetics is controlling that trait. And you can see that the answer to that question is that it is quite a lot. So over 50% of the variation in the traits that we measured, and these are field-grown trees, of course, is due to the genotype of the scion. And so for me, this is really exciting because it means that we can use this information and data for breeding. All right, so what about the rootstock? What does that look like? And so the answer to that is shown in this plot here. So So it's the same setup. X-axis is the traits that were measured, y-axis is the amount of variation that's explained, in this case by the rootstock. And you can see that it looks quite different. So for many traits, the rootstock contributes very little to the variation that we observed. There are some notable exceptions on the right end of this plot. So brakes and acid in particular seem to have a large contribution from the rootstock. And now that we know this, we can account for it in some of the models that we're building. Okay, so the citrus variety collection is grafted mostly on Carrizo and C35. And so we wanted to understand if this relative contribution from the scion on the rootstock was similar in other situations. And so we used a lemon trial that's located in Riverside, California. So this trial consists of 7 scion genotypes and 3 rootstocks. And the plot setup is similar. So the x-axis is trait, this time measured, traits measured by drone imaging, so it's related to tree size and architecture. And then the y-axis again is the variance that's explained by either scion in blue or rootstock in gray. And so you can see the trend is similar. The scion genotype is explaining more of this variation that we measured than the rootstock. Okay, so the ultimate goal, for me at least, is to understand not only that the scion or rootstock is controlling these traits, but what particular regions of the genome are associated with those differences. So I just want to remind everyone that we're looking for places in the genome where, like in this example, we have a change from a T to an A that I highlighted in gray. And I'm going to ask, can I find positions that explain differences in the fruit that I've observed here on the right? All right. And so the example that I'm going to show you today is for one trait in particular, so I'm going to focus on seed number, okay? So here what I'm showing you is the distribution of seed number for various different genetic groups or market groups in the citrus variety collection. And so the y-axis is telling you how many seed on average those particular groups have. So on the right end of the plot, you can see that citron and pomelo and trifoliate and other relatives have very high numbers of seed, while on the left side, navels and sweet oranges and the like have very low numbers of seed, as we expect. Okay, so we're going to look to see if we can find regions of the genome that explain these differences that are shown here. And so this is the way this type of analysis is typically visualized, and again, I'm going to walk you through this plot. So every point on this plot is one of those AT variants. So it's a genetic difference that we've measured in the 600 lines that we're studying. The x-axis is the position on the citrus genome that variant is found, and then the y-axis is telling us how associated is that variant with seed number in this case. So the higher a point is, the more associated it is with our trait of interest. And so I want to draw your attention to this region on chromosome 1. It's a little hard to see on these projectors, so I apologize for that, but there's a region on chromosome 1 that's associated with seed number. And so I'm going to show you some examples of what that difference looks like. So using that cartoon example of the AT variant, what we found is that, like, let's say A, for example, is associated with high seed number, while T is associated with a low seed number. And examples of pomelos that have those different genotypes are shown here. So you can see the one on the top has many more seed than the pomelo that's shown on the bottom. And I find that this high allele is enriched in groups like citron, pomelo, and trifoliate, and the low allele is enriched in mandarins. Okay, so how can we use this for breeding? So if we're breeding scion varieties, perhaps we are interested in having a low-seeded hybrid, so we can use markers to select for that low-seeded allele. And perhaps in the opposite side of that is if we're breeding for rootstocks, maybe we want to select for individuals that have a high number of seeds, and so we would select the high allele that I just showed you. And so I just used seed number today to illustrate this point, but we can find these associations for all of the traits that we measured on the pack line and in other phenotypes that we're interested in to identify regions of the genome that are associated with those traits and enable our program to select hybrids at the early stages and ensure that those traits make it into germplasm at the end of the pipeline. Okay, so I'm briefly going to talk about how we're going to use the same type of strategy to identify the genetic basis of tolerance to HLB in the field. So this work is just getting started, so it was recently funded in a collaborative grant between myself, Kim Bowman at the USDA in Florida, and others. And so what I just walked you through, how we measure traits and look for association, is going to be used in this situation as well, except for the phenotype is tolerance to HLB in the field. So the material that's being used is 15 replicated field trials that were initiated by Kim Bowman. They were planted starting in 2014 in 3 growing regions in Florida. And this work can really, it's important to note, I guess, that these field trials are uniformly infected for HLB. And so you can really get at the genetic basis of tolerance to disease in this situation. So across these field trials are 350 novel rootstock hybrids that are highly replicated. So this includes a total of 6,000 trees, and field performance has been monitored annually in these trials. Okay, so here's some preliminary data that is hopefully illustrating how exciting this material is. So this is the field performance of 80 hybrids grown in 2 trials. And on the x-axis, what I'm showing you is the cumulative yield over 3 years for these 80 hybrids. And the y-axis is tree health. And what is pointed out on this plot is some HLB susceptible industry standard rootstocks. So it's hard to see the colors, but in red, pink, purple, and greenish. So those are pointed out on the plot. And then a commercial HLB tolerant rootstock known as Super Sour 3 is shown in blue. And so what I want you to see is that there's a number of hybrids highlighted in green that have performance that already exceeds industry standard HLB tolerant rootstocks. And so this is only the first 80 of those 350 hybrids. And so the goals of this grant are twofold. So it's to identify and release these best performing HLB tolerant rootstocks. And this material is already making its way through the CCPP, so it'll be available in California once these varieties are released. And then the second goal of this work is to discover the genetic basis of this field tolerance to HLB. And so if we can identify regions of the genome that confer tolerance, we can use it in our breeding pipeline to identify new hybrids that have these loci of interest to ensure that the material that's going into the downstream end of the pipeline will be HLB tolerant. Okay, so today I hope that I was able to show you how work in the core program at UC Riverside is aimed at increasing the efficiency of hybrid selection. And so far we're doing this in 3 ways. So we're confirming the parentage of hybrids to remove those nucellar seedlings. We're implementing automated phenotyping approaches to develop vast catalogs of phenotypic information in citrus, and then we're using this information to facilitate trait discovery in both scion and rootstock material. Okay, so with that, I'd like to thank all the folks that contributed to this work. So that includes members of the core breeding program, which is Mike Roos in his group, Tracy Kahn in her group, Peggy Mock, and Glenn Wright. The folks in my lab are listed here as well. And I'd also like to thank Ashraf and his staff at Linkove for facilitating the pack line analysis, and the funding, of course, from the Citrus Research Board, the USDA, and UC Riverside. So with that, I'm happy to take any questions.
We have a question over here in the front.
I didn't know if there was a mic coming or
There's
not.
one sneaking up behind you.
Thank you. Thank you for that presentation. The stuff with the rootstock is exciting to see that is being developed. I was curious, is the same procedure now being implemented for science stocks so we can do best matches?
I am not sure that I understand your question. So what do you mean by the same procedure?
So you went through and looked for the best HLB resistant rootstock. Now we're looking for the best HLB resistant science, and hopefully we already have those hybrids made in pots somewhere that are going to be rapidly expanded.
Yes. So this type of work that I presented to you can only really be done at a large scale in Florida at the moment because of the number of trees that need to be evaluated to identify those best performing rootstocks. So certainly I imagine that work is ongoing for scion breeding, but it's going to be carried out by the scion breeding programs that are located in Florida, so I can't comment much further on that.
I would maybe speak up a little bit and say we do have Mike Bruce over here who's doing some scion work, and I think there's some work going on at a BSL-3 facility down in Riverside to test some of the California varieties for HLB tolerance.
Thank you.
So, are there any other questions from, oh, we've got one up front.
With the resistance genetics that you observe in the scions, with that for the rootstock, would that also hold for the scions? Is that known? Is it, there are certain traits that were more predominant from the rootstock than from the scions. Does that have anything to do with resistance to HLB?
So in those plots where I was showing you the amount of variance that's due to scion and rootstock, this is in the absence of HLB, okay? And so it's like sort of dicing up a piece of the pie, right? So it depends what that pie looks like. So if we put those varieties, let's say in an extreme environment, then more of the variation might be due to the environment than genetics. And so this is in flux, right? So that's why I tried to show you a comparison to see if it's consistent, but if it's not always going to be the exact same answer every time you perform those measurements. And so I think it's exciting because if you're interested in genetics, if something only explains 5% of the variation, it's going to be a very, very difficult problem to study, and so we're not in that situation. In terms of whether people have seen this before, I think this is one of the largest phenotypic data sets on so many different varieties that's been collected in citrus, and so there's many trials where this has been addressed, but this is usually just looking at a limited number of individuals. Mike?
Yeah, I just want to quickly.
Well, I'm not sure if Rick Danzler is going to talk about this later, but there is a kind of parallel project on finding resistance genes in scion varieties in Florida. Okay, so there is an ongoing project where there's a bit of a struggle among the researchers and funding agencies to fund this project, but it is going forward at the moment.
When you're looking at the traits, are you finding more the promoter regions are affecting the outcome, or is it the variation within the genes themselves?
All right, so I can't see who's asking the question, but there we go. So the density of our markers is not quite high enough yet to really zoom in on what change is causal, right? So we're just getting sort of in the neighborhood, and so it's a little too early to tell if these changes are in genes or promoters or other areas.
Do you believe that this work can be utilized to determine or predict incompatibility or compatibility between scions and rootstocks?
I would love to work on that problem. I think the major challenge is going to be the phenotype. So how do we measure and quantify what incompatibility looks like in a consistent way? And if we can do that, then of course we can use these tools to address that.
Okay, one more quick question from Rick.
This is not a question, but to follow up on Mike's point. I'm Rick Danelle from Florida, and I'll be speaking this afternoon about some of what we have going on in Florida. But there is a huge scion top working trial that is underway where we asked the University of Florida and USDA breeders to give us their top scion candidates. They were inserted into rootstocks based on top working, so those trees are fairly old. We're going to hopefully be able to get a reading on how well those new scions do more quickly than if we were putting a new tree in the ground. CRDF is also in the process of putting in a new scion trial, where again we ask the breeders that we help fund to give us their most likely candidates for success. Those trees are going to be planted in about 18 months. It's not something that's going to happen right away, unfortunately, but there is a lot of scion work underway that I think feathers very nicely into your presentation, and you and I need to speak some more.
Happy to do so, and apologies if I misinterpreted that original question, misstated some of that information.
So I see Tracy has a question. If it's a quick one, but we're going to need to get ready on the next presentation. We've got Holly right behind you, so a quick comment.
Okay, so I just wanted to follow up on the last comment, which is that, and I think this was pointed out, and there is actually a poster that combines Florida breeders as well as our core project that Danelle just talked about bringing in these, and the first 19 of the different scion and rootstocks that were brought in through the Citrus Clonal Protection Program are now through quarantine, and they will soon be planted at Lynn Cove so people can start to see, how they perform a little differently than in Florida.
Thank you for that. Tracy, we do have a lot of great posters out there, so that's another one hopefully you'll be able to see either at lunch or at the afternoon break. But with that, I'd like to thank Danelle again for her presentation.